Welcome back, everybody. This is going to cover chapter 18, which deals with diagnosing infections. All right. Here are the learning outcomes for this chapter. Again, if you want to test your knowledge and make sure you're learning what you're supposed to for this chapter. So trying to identify unknown bacteria. Your patient comes in, they're sick with an infection, and how do you figure out what's causing it? So there's these three sort of main categories of methods that we would use to try to determine that. That's the phenotypic methods, the immunological methods, and the genotypic or genetic type of methods. There are some variable ones, some random ones as well that we wouldn't really put into these categories. We've also have an increase in point of care diagnosis, which means running the tests at bedside and getting the results a lot sooner so we can diagnose infection sooner and start treatment sooner. So that's been great. And they can even be performed by patients at home, and this does include your at-home COVID tests as well. So that's helped out a lot with keeping control of the pandemic and not letting it get too out of hand in areas. So that's been helpful. All right, anytime we're talking about phenotypic testing, we're talking about observing a microbe under the microscope, looking at the macroscopic aspects of them. So especially if we're talking about mold or something where you can actually see some aspect of it, or even symptoms where you can see discoloration of the skin and or redness, anything like that. Morphology of the organism if you were to culture it. Physiology of the organism, that would be including certain biochemical properties, so they would actually go hand in hand, but depending on the organism, of course. But for bacteria, physiological and biochemical properties basically go hand in hand. And then antimicrobial sensitivity as well. Then we have immunologic type tests. We're going to analyze the microbe utilizing the power of the specificity of antibodies and antigens. And then genotypic testing, where we're going to be looking at the actual DNA or RNA of the microbe to get a more genetic-based answer. So here are some sampling sites on the human body that we would obtain our samples for testing, all the different tests we're going to be talking about. You can see there's a variety, and most of these are quite familiar with you. I don't know. Blood samples, urine samples, stool samples, and basically anytime you're getting a swab type specimen, you might want to put it in a specific transport type medium. We had introduced the concept of a transport medium in the tools of microbiology chapter, but this is the idea of what it might look like. You have your actual swab inside of this a holder, this case that keeps it safe, so it's not going to get damaged, and it's also not going to contaminate anybody else. You have media in the bottom of it that helps maintain the health and stability of the organism until we can get it cultured properly. And then you can also squeeze on the bottom of it to help mix the microbe sample in once you've swabbed whatever thing you're swabbing. And whatever, it all has, and it has the tamper evident cap, which is also important, right? You want to be sure you're not swabbing somebody else with another person's swab. So that's our transport media. Specimen collection. After we have actually collected our specimen, we're going to transport it to the lab, and they're going to store it, usually refrigerated, until they're going to culture it, or they're going to save it in case the culturing mechanisms they're using don't work, whatever it is, but they usually won't get rid of it until there is some sort of consensus on a diagnosis. Right, and so it can be maintained in the transport medium and then later into the refrigerator for hours, even days, depending on how it's treated. Sure. So you might want to consider what I was saying on the previous slide, what you might want to consider how it might need to be treated if we're talking about an anaerobic organism. If you are suspecting something like gas gangrene, you wouldn't want to expose it to the air for very long, so you might want to make a special note of that. So you will need to, as a healthcare professional, know how to properly collect specimens, transport them, and store them, label and identify them when you pick up a specimen. You want to provide an accurate patient history to go along with your order for whatever testing you're giving to the lab. And most of the time, a lot of these orders are going to be submitted through something like Epic or an actual like online like network type system nowadays. So, but this is typically the type of sheet that you would see associated with it. You're ordering a test from microbiology. Maybe this person is coughing a lot, so you send a sputum sample and you want an acid-fast smear done, or you could even order the culture straight up if you just want to go ahead with that. And then you'd have to wait for culturing and for it to grow, which, by the way, mycobacteria are notoriously slow to grow. But once it did grow, you could then do the acid fast on that if you suspected there wasn't enough bacteria in the sputum sample, or you'd want to be sure you didn't miss anything. So anyways, that's how you would look at something like this, and then you would get your results back from these guys as to whether it was acid fast or not. And being a medical professional, you might want to understand what those results mean. So there are specialized media to help identify these organisms. We have the selective media and the differential media, which we've talked about previously. So selective media, we can use this to enrich the specimen. Some things will be able to grow and some things won't. So if we suspect that it is a gram-negative enteric that's causing the problem, something like E. coli, we might grow it on like a EMB or McConkey agar, something that will favor gram-negative organisms and even enterics in comparison to gram-positive organisms, which could be infiltrating the sample as well. So that could help us achieve isolation even faster. Differential media, we could then use that to help identify physiological sort of characteristics or ability, fermentation patterns, stuff like that, based on the color changes associated with that media. So all of that biochemical media that we're going to be talking about here in a moment would fall under differential based on how that organism behaved on that media, created whatever byproduct, and caused that color change in the media due to the presence of that byproduct. So this is a dichotomous key as you collect data, as you're trying to figure out what your organism is. You would maybe start with your gram stain. Is it gram positive or gram negative? Let's say it's gram negative. Is it aerobic or anaerobic? Let's say, how do you want to say? You might test whether it is oxidase positive or catalase positive, which are indicators of aerobic activity. Let's say it's anaerobic, and then that would lead you to, these sets of organisms, and et cetera. And this is how you could get to the identity of your organism eventually. So you could get to a broad answer, and you might need to do more testing to get the more specific answer, but now you might have a direction to move in, whereas it might have been too daunting, having not already looked through that dichotomous key sort of setup. So biochemical testing, here we're looking at those physiological reactions of bacteria to the nutrients in the media. And that indicates certain enzymes being present in bacteria, and not all bacteria are the same. So based on their reactions and visible color changes, we can say that they are positive or negative for being able to break down or utilize whatever molecule it is that you're testing. And that will help you define even more what organisms you could be dealing with. So nowadays, biochemical testing can be performed by machines or manually. If you are really looking at an actual lab that's going to be testing a lot all at once, you might need the machine version. But if you're not doing this very often, you might do this manually. And what you would do is you would culture your medium in an assay medium, right, made for the specific assay. And then you can put drops of your culture into these little bubble areas in here, and then incubate them so that the bacteria could grow. And if they can utilize whatever that substrate is in that little pocket in there, can lead to certain changes of color in that. And then you can compare the color changes to a key basically, and sometimes you would be able to have a machine look at it for you, but it just depends on what system you're using, of course. And anyways, you get the color change happening to whatever is going on in that little pocket, and you mark down whether it was a positive change or not, and then type in a little code that corresponds to that positive or not, and that will give you the answer for what organism it should be based on those results. So it's like doing all those biochemical tests. Each of those pockets represents a different biochemical test. So it's like knocking out a whole bunch of them all at once in one sort of sitting, which is way easier than doing it day by day by day. All right, antimicrobial susceptibility. We would use this, of course, to determine what drugs we might want to use in treatment, which is useful in and of itself. But you also might be able to use this, depending on the organism you're concerned about, be able to use this to identify your organism. So if we look at Streptococcus species, Clostridium species, or Pseudomonas species, these types of bacteria will have specific patterns of sensitivity to certain antibiotics. So that can lead us to a presumptive identification or genus identification as well. Looking at phage typing, this will involve viruses that attack very specific bacteria. They can be species specific, and they can even be as specific as strain specific. So we can use this for identifying the different kinds of salmonella that can cause disease, like Salmonella typhi versus Typhimurium versus the Enterica, all of those other ones. So if we were looking at whether or not that phage could infect that particular salmonella or not, could tell us what strain of salmonella it is, which is pretty interesting. So you just create a lawn of growth that just covers the entire plate, and then you dot it with phage that would be able to infect salmonella, and or dot it with all sorts of different kinds of phages to all sorts of different kinds of strains of salmonella or other organisms that would be gram-negative, whatever it is that you're doing. And then if there is a plaque that forms, which is just a clearing in that growth, because that's going to kill bacteria when you expose it to the viruses that infect it, it creates a clearing. So wherever you see a clearing, that is where that phage is that can infect that particular bacterium that you put all over as a lawn from your patient. So that's pretty cool. That's called phage typing. All right, clinical significance of cultures. We want to determine, okay, so let's say you take a sample from a patient and you get some growth, and you want to know if this is just a contaminant or if it's normal biota or if it is actually clinically important. All right, so if you see on your media, maybe just a couple of colonies from your sample streak, a couple colonies of E. coli, you ran your test and it looks like just a couple colonies of E. coli, that might make you be like, it could just be from normal biota or exposure to normal biota. But if you have hundreds of colonies of E. coli on, an area where it shouldn't be, then that would be indicative of the infection then. But there are some bacteria out there that there should not be any of them on your body ever. And that includes Mycobacterium tuberculosis in this example here. It is a true pathogen, and if it is there as a pathogen, and it should not be there. So even one of these organisms being seen would indicate an infection and should be taken seriously. We're talking about the phenotypic analysis of these organisms, biochemistry, growing them on these different kinds of media and seeing how they respond, all of that. So culturing microbes takes a minimum of 18 to 24 hours, depending on the organisms you might be looking for. If you're looking for, some are often a lot longer, like it says, like Mycobacterium tuberculosis, you could be waiting over a week, even 2 weeks for it to grow up to a usable level of bacteria. So so many conditions can be caused by organisms that you can't even culture as well. So this is all causing a problem with our ability to now diagnose based on these phenotypic methods. So what are some other things we have available to us? We'll get to that in a moment. First, I want to summarize our phenotypic methods. We have our direct examination. Direct examination, that would be like looking at the organism or slides or stains of that organism. We have selective and differential growth. So growing on media that would select for that organism, or growing on media that could change color. That often relates to biochemical testing as well, changing color due to the presence of enzymes breaking down things in that media and then causing a color change as a result. Susceptibility testing, dealing with the antibiotics, and then other ones that we talked about were phage typing. There's also animal inoculation if you need to inoculate an animal to grow up your bacterium faster or to mimic the disease in an animal. You might be able to do that with certain strains that you wouldn't be able to culture otherwise, but that is an option. And then we also have cell culture growth. So if you're trying to grow up, viruses, you can't grow viruses by spreading them out on a plate just by themselves. That's not how viruses live. They have to be inside of cells. So you have to give them cells to grow inside of. So you might have to generate, tissue culture is what we would call it, in order to check out the viruses. All right, so next is some stuff that isn't phenotypic and you might move on to next, which is the immunological methods, including serological methods. So serology deals with testing serum of a patient. So you allow the blood to clot, you separate the clot from the liquid. The liquid is the serum, and you have antibodies in that serum. So those can be tested to see what they bind with. You might also have antigens in that serum. So depending on what kind of testing you are doing serologically, you might look for the antigen or you might look for the antibody. It depends on what you have available as your known thing to test it against in order to figure out what this organism is that's causing disease, right? So you can't just say, oh, let's do an ELISA on this sample that I think is, Ebola, and you don't have any antibodies or antigen from Ebola to test with. How would you do the ELISA? How would the ELISA work then? It can't, because there's nothing for you to test it against. So that's just an example. Right. So you have to have a known in these aspects in all of these tests here. Okay. So remember, antibodies have extreme specificity for the antigens that they bind to. And then we can utilize that to lead to color changes and all sorts of stuff to tell us whether or not we have binding of antibody to antigen. And if we have antibody binding to antigen, then we have a positive test. So unknown antibody can be detected using a known antigen, or unknown antigen can be detected using a known antibody. You have to have one or the other. You have to in order to do serological testing. So nowadays, you don't only use serum in serological testing. We have sophisticated enough equipment that we can now also test not just serum, but urine, cerebrospinal fluid, whole tissues, and saliva in similar tests. So here's the basic principle involved in serological testing. You have your patient serum. Do they have the antibody? Well, if you don't know if they have antibody, well, then you better know the antigen so you can, see if it's going to bind. So you give it the known antigen and you mix it with the patient's serum. And then if you see this sort of a thing, we have this agglutination. This is just basic concept, okay? We see agglutination or clumping up, and that means antibody is binding up with antigen, causing the clumping. That is an example of agglutination there. So if you want to know the identity of the actual microbe, let's say you took a sample from the patient and you have isolated a colony, but you don't know what it is. So you think it's salmonella, or I guess in this case, it's specifically Staph aureus, staph infection. So we have known antibody here. We mix the two together, colony with the antibody, of course, in a solution probably. And if they are interacting, again, we will have agglutination and clumping of that sample. So it allows us to determine what's going on there. So there's all sorts of different ways we can utilize this concept, whether it is agglutination or precipitation. Those are very similar concepts. In agglutination, we have antibody-mediated clumping of actual whole cells. In precipitation, we have smaller complexes. That's just antibody-antigen complexes. But cells, clumping of actual cells is agglutination, otherwise it's precipitation. So to accurately describe the slide that we were just looking at previously, would in fact be precipitation, right? To make that clear when you guys are going back to study. So immunochromatography, we have some sort of color changing thing if there is the presence of an antibody or an antigen, whichever one you're looking at. So turns out, well, we're going to get into this. We have Western blot, which uses electrophoresis to separate proteins, just like we did with DNA, except in this case, we're going to use antibodies or antigens to determine the identity of whatever protein we ran in the gel. We have fluorescent antibodies that literally are going to be antibodies that fluoresce under certain kinds of light, and that if they do fluoresce, that means that the thing that they're binding to has to be there, because we would have rinsed away in rinses. Then we have ELISA, which we learned about in lab. We have a sandwich technique, or we have the indirect technique, either looking for the antibody or looking to see if antigen is there. And then we have in vivo tests, looking at reactions in the skin itself. We'll get to all of these. So antigen antibody interactions, again, antibody is going to associate with a particular, very specific site, very like exact chemical structure and makeup, which we call an epitope, which is present on an antigen, which is something that we would be reacting to with our immune system. So you can't actually see this directly. So you can look for color changes that we can accompany this thing happening, or we can look at clumping or something to see if there's a change in the solution or mixture of cells or whatever it is that we are looking at. So the clumping aspect, we're either looking at the cells clumping in the case of agglutination, or the antibody antigen just complex just binding to each other, and that would be precipitation, right? So we just said that. Often we can be using red blood cells to see if they agglutinate or stick together, which is what we do for blood typing. So if that was the case, and you see the blood cells clumping together, for example. So you can do all sorts of stuff like that. Precipitation, though, we're looking at the actual soluble molecules. They're going to be very tiny, but you will see precipitation associated if you do have the reaction, where you wouldn't see it if you didn't. So for either of these, we have antigen antibody concentration needing to be optimal, allowing antigen to be interlinked by several antibodies to form that clumping. Otherwise, if there's not enough of that ratio between the two, you're not going to see the clumping, and it won't be very useful as a result. So it's not super sensitive that way, you see, because it won't pick up every single antibody or every single antigen that could be present, but it could be, so it's not super sensitive, based on my point. Okay, agglutination reactions. We get the visible clumps of cells. We use this for blood compatibility. This is just showing a picture of how that might work. If you have, agglutination, we have sticking together of these cells, and you would be able to see that in your result well, compared to the normal pellet that would form whenever your cells aren't clumped together and associated with antibody reactions. All right, immunochromatography, we mentioned just a second ago. Here we're talking, these are like lateral flow tests. Okay, and you do know these, and you know exactly what I'm talking about. These are at-home rapid tests. We can find these in the drugstore for pregnancy and for COVID-19. We've seen these before. They are plastic cartridges. There is basically some sort of medium, whether it's like a cotton-based strip or something. There's a strip along which a liquid would be able to migrate, and as it encounters, the bars on there, which actually might contain antibodies that we know the antibody, or it could be antigens that we know the antigens in the case of other situations. But anyways, there's still antigen antibody reaction going on that will lead to color change if there is binding there. So that's immunochromatography. Think of anytime you see chroma, think of color. So this one is going to lead to color change lines. Immunochromatography is still dealing with immunology driving this whole thing. So if we're looking at antibody titer, this is dealing with the concentration of antibodies in a sample. So determining how many antibodies essentially this patient has to whatever thing that it could be, you could be infected with. So we'll dilute out their serum, and the dilution where we still actually get agglutination or precipitation, depending. The lowest one, that would be, the highest one that still produce the agglutination, that would be what we consider to be like the titer or the measure of how much antibody there would be in there. So So highest dilution. Now, when I was saying the lowest, that is the lowest concentration. That, okay, you understand the difference, right? Highest dilution means we've diluted this 400 times diluted, and would be normally to 10. So this is our first one, so that's the 10 times 10 dilution, our times 100 dilution, our times 1,000 dilution. If we're still having reaction at the times 1,000 dilution, diluted 1,000 times over value-wise. So we did 1 to 10 dilutions in every single step, and we're still getting agglutination at the 1,000, but not at the 10,000, then 1,000 is the highest dilution that we're still producing agglutination, but it is still going to be the lowest concentration that we're still seeing. It's the same thing. That's what I'm trying to say. So be sure you understand that concept and don't get confused by that. All right. Right. So antibody titers, what do they do with these numbers? They use this to keep track of autoimmune diseases. If you have anti-self antibodies, for example, and past exposure to diseases as well. So if you've seen the thing, the antigen, the pathogen recently, then you will have active antibody in your blood, and we can titer that to see how recently we think that could be based on the concentration. So serotyping. Serotypes are bacterial subgroups. We have antigen antibody technique for identifying, classifying, and subgrouping certain bacteria into serotypes. Sometimes they might have certain characteristics, and sometimes it'll be hard for us to tell the characteristics, but we call them serotypes because we can identify them based on these interactions, so antibody antigen type things. That will help us look for the presence of certain flagella, whether it has flagella or not, whether it has a capsule or not, and certain aspects, characteristics of the cell wall or not. But these antibodies can tell us the differences between these organisms, so we can tell what's what. Another immunological based test, we have the Western blot. Samples of proteins from bacterial cell or virus. We will run them out on a gel, just like the DNA gel electrophoresis. The smaller things move faster, the bigger ones stay up at the top. And we're looking at proteins and not nucleic acids. Then we will move the proteins out of the gel using more electricity in a different direction onto a little piece of paper. It's not paper, it's nitrocellulose, but it looks like paper. It's a special membrane. And then we will expose it to the patient's sample, like serum or something like that. And then we will look to see if there's bands on that membrane. So it's a little bit more complicated. Here's our gel here. We'll put the membrane here. This is what we're interested in. And you'll put on each side blotting paper just to stabilize everything and protect everything. So we can get movement using electricity from the gel and onto the membrane. Now we've got it on our membrane. We've got proteins here. We know there's a ladder here, which we could potentially be able to visualize right up front, depending on what you used, but we can't see anything with our proteins yet. So what we do is we block first, and that binds with every single blank area on here, so we don't accidentally get a positive when we shouldn't. Okay, so we've blocked everything. And now we put the patient's serum on there. And if that patient has antibody to that protein, we'll see it bind at that band there. So we just soak it and let it sit for a little bit, do some rinsing, and then we'll do antibody. So that's our secondary antibody, just like we do with indirect ELISA, that has some sort of enzyme or some sort of other marker on there, so that we can look at that by exposing it to a color changing thing or whatever, and take a picture of it, and we can say, yeah, that's such and such. This person has this protein, which means that you have the antibodies against this protein. So because they have antibodies against this protein, they have recently been infected with that protein, virus, etc. Okay, so immunofluorescence. So we have a direct and we have an indirect with this guy. So direct, again, concept is, does the patient have the antigen? We have our specimen, our test, our sample from our patient. And then we will put that onto a slide. It just works differently than ELISA, but we're still just looking for antigen. So we'll put our sample on a slide and expose it to a known antibody. Okay. Our unknown, we've put it on a slide. we put a known antibody on there. And if they do bind, and that special known antibody fluoresces when it's exposed to a certain color, a wavelength of laser, then it'll glow and we'll know that there had to be binding going on. Okay, so we wanted to know, does the patient have the antigen? And if they do, then it glows because we have a fluorescent antibody. Fluoro, that means glowing. If it's indirect, does the patient have the antibody, not the antigen, the antibody? So now we have a known antigen. You have to have one or the other. Patient serum, we will we'll have antibodies used to recognize the FC region of antibodies in the patient's sera. That means that if there are antibodies in the patient's serum, then they have antibodies that will react against the fact that those are antibodies. Okay, so we have to have that. So we have a known antigen. And then, so we have the known antigen on the slide. We add our serum from our patient, and then we bind our secondary antibody that recognizes that there are antibodies there. So a whole other antibody does that for us, and it's tagged with a fluorescent molecule of some kind. And so that will, if there is glowing in this case, then we know that the patient has the antibody. So that's basically how that would be working. So antigen, we know what it is, put the patient serum, and then we treat with the antibody, secondary antibody is what we call that. And it is fluorescent. And if it stays there after the washes, that means that that secondary antibody bound to an antibody that bound to the antigen. And that antibody in the middle, that had to be antibody against the pathogen of interest. Okay, we know about ELISA. We have an enzyme linked indicator that will allow us to see if there is a reaction going on there, which leads to color change typically. And it also relies on those plastic wells being able to have things stick in the bottom of them. And so we can do all the rinses and have that work out. So indirect, does the patient have the antibody? Same concept here. So what we're going to be doing, we're going to determine if the patient has the antibody. So we have a known antigen, we'll adsorb it or stick it to the well, and then we will add the patient sample, patient serum. So this known antigen, add the patient serum, and then we can do rinses. That complex, if it is there, will stay there after the rinses. All right, so we've rinsed all the excess so we won't get some sort of false positive. And then we add a secondary antibody, again, an antibody that binds if there's an antibody there. And this will have an enzyme that will cleave a substrate to lead to a color change. So color change is positive in these. So this is that step-by-step look at an indirect ELISA. I'm not going to walk you through this one because you guys had this in the lab, but you are welcome to look through it and read through the details. It's just exactly what I said to you guys just now, but with pictures and I don't know, clearer words. It doesn't stumble as much as me. So our sandwich or direct ELISA, does the patient have the antigen? So why is it sandwich? Because we're going to sandwich the antigen between the two antibodies. And if you're positive. So we have our known antibody, and we'll stick that in the well, adsorb it into the well, and then we will treat it with our patient's sample. Patient sample gets mixed into that. So if a complex forms, if it is positive, we'll rinse that, and that'll stay there. So it'll be known antibody and then antigen hanging out here. And now we'll add a secondary antibody. This time, this antibody will recognize that antigen again and have that enzyme that cleaves the substrate that leads to a color change. And that's nice. And so that would be positive again. So same result here. So this is just stepwise look at that. Here we have antibody. Here we have added the patient sample. So it looks like this patient has the antigen of interest. So antibody bound its antigen. We add our special secondary enzyme linked antibody that also recognizes the antigen. We add our substrate. The enzyme cleaves it and gives us a color change, positive. So this is an example of how something like an ELISA would look in an actual medical setting. You would be also doing everything in at least duplicate. Every location has its own regulations on that. So you would see a repeat positive, 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 instead of like, just dots all over the place, random. So you would want to have the same patient tested repeatedly in these because they are not particularly sensitive. So, okay, moving on to the complement fixation test. This is just utilizing the concept of complement and its ability to break open cells with the membrane attack complex they can form. Utilizing that to see whether or not patients have antibody. But how? How does that work? So essentially, if you do have antibody against whatever pathogen, basically, we take your serum, you have the antibody, we mix in our known antigen. So I don't know, COVID viruses getting mixed in with your blood sample. All right, if you actually have the antibodies here, we'll get binding of that antigen with the antibody. That's important because you don't see that here, right? So in step 3, we will add in complement. If you've had binding of antibody to antigen, forming those complexes, the complement will bind to that. And that's great. But if you didn't have that happening, which we didn't in number 2, down in the bottom down here in the lower part, the negative test, we're just going to leave the antigen and the complement floating around in their own free will. There's no antibody to bind it up. If the antibody was able to bind it up, then essentially what we have here in number 4, we have distracted the complement, so it will not poke holes in red cells that we expose it to. They'll be fine. So we'll have intact red cells here. However, if there was no antibody to bind up the antigen and the complement proteins, then when we add red cells, no antibody to bind it up down here. So when we add the red cells, they're going to get lysed or broken open. And so there's no pellet in these ones. So that's the idea of how complement fixation can be used to test if you have an antibody against certain diseases that would be associated with complement activation. So viral and fungal diseases, it says, are often associated with that sort of reaction. Right. In vivo testing here, this is pretty similar to serological tests. We're looking to see if there was a reaction, if you have a T cell response, if you have basically an immunological, there we go. If we have an immunological response to tuberculin protein, okay? And if you do have a reaction to it, it means your body has seen it before and you are likely positive for tuberculosis if you do test positive. So that's how the concept of how that works. It's in vivo because it's actually in your living body. Okay, so we come back to that concept of specificity versus sensitivity. Let's go through these again. Specificity, we will focus on only a certain antibody or antigen. Only that certain thing and not anything that looks close to it or anything, just only that specific exact thing. Doesn't react with unrelated or distantly related antigens. So it will only react with this thing that you're interested in. So it has high, so if you have high specificity, where you're only reacting with the thing that you're interested in, you will have a low false positive rate because you won't be reacting with things that aren't the positive. However, you risk false negatives as a result of that. With sensitivity, the detection of even the smallest minute quantities of antibody or antigen, depending on the test, can be detected. So any sort of test that has high sensitivity, pick up even the smallest amount of these things. So it's going to have a low false negative. You'll pick up on the positives really easily. However, you risk false positives because you could potentially be overreactive. Potentially, okay? All right, let's move on. We also have, after the immunologic, now we have the genotypic diagnostic methods. So you can either do this via amplifying the nucleic acid, making more of it, hybridization, binding basically probes to certain sequences of nucleic acid. And then we have whole genome sequencing where we have certain basic sequences that will be able to bind to during the PCR that allows us to sequence a whole genome very, very quickly and very cheaply nowadays. Okay, sorry. Is it chain? All right. Polymerase chain reaction, PCR. It's used in many nucleic acid tests. We can have numerous copies of DNA or RNA within hours. This can amplify even small amounts of nucleic acid in a sample. So it's very sensitive for diagnostic testing. It can be performed on bacteria, viruses, protozoa, and fungi. So it's pretty broad spectrum. There's all these different kinds of PCR, real-time, reverse transcriptase. If you needed to test RNA sample, you would use the reverse transcriptase because you want it to be DNA for the actual PCR testing. We have multiplex, we have pan-bacterial, we have RT lamp versions. The most commonly, I feel like biggest main one that people use anymore is real-time PCR. So it's going to actually allow you to look in real time to see if you are having elongation, basically, as a result of the specific primers that you put in there by giving off. Usually it's a fluorescence that the machine watches while it's going on with its little thing. So it's pretty cool. All right, hybridization. This is using probes. So little pieces of DNA, small fragments of DNA or RNA that will be complemented to specific sequences that we associate with a particular microbe. And we can, essentially take a sample and heat that sample, and so it'll spread the DNA apart, and then pop it with these probes, mix it up with the probes and rinse it, and then look for binding. And how we do that depends on the test that you're running, but but that would indicate that you are looking for the right microbe if you found binding there. So that's how it's designed. So fluorescent in situ hybridization, FISH. Here we have fluorescently labeled probes that we can mix with intact cells from a patient or an environmental sample, depending. And then we can locate glowing cells, because if we have hybridization with the probe that looks for that specific microbe or whatever, then it will glow with fluorescence and allow us to determine if that organism is present or not. So this is just showing us how we might identify staph with such a thing. So this is a different kind of a specific kind, of FISH. So this is a peptide nucleic acid FISH testing, but you don't really need to worry about that part of it. So essentially, we have our staph on some sort of substrate, like a slide or something, and we have our probe that is fluorescently labeled. And if it binds in with that sample, then and it is then it will fluoresce, it'll glow, and then we'll know that there is staph there because it's specific for staph. And you might not do this right off the bat, like this is showing you. You might do gram stain and see that there are, cocci that are, in irregular clusters. And you might think it's staph, and it's stained positive in gram stain. So you might think it's staph. So you go on to do this next to confirm it. That's how that might be used. We also have the availability of whole genome sequencing. It's pretty cheap and it's pretty commonplace nowadays whenever you're working with things that might be a little less familiar. So this is also good for rapid analysis of outbreaks and drug-resistant organisms to learn more about their genome and what makes them tick, essentially. We can also use what's called deep sequencing, where a single genome can be scanned and analyzed multiple times with fewer errors, but that would allow for more accurate data to be collected. Compiled is a better word for it. All right, microarrays. These are essentially plates or chips, usually with a ton of different things on it. It could be proteins or it could be nucleic acids. In this case, we're talking about nucleic acids. All laid out on this chip. Then you can go and mix the patient's sample with that chip surface, and based on which one's positive might indicate that you have this disease or that disease or whatever. So it's essentially what would be going on with a microarray. Patient sample or nucleic acids isolated from the patient are incubated with labeled gene sequences on the microarray. And if you have matching up of the sequences, there will be fluorescence again that the computer can detect and then tell you whether you're positive for, row A, column C or something. And then because of that, you are positive for Staphylococcus aureus. All right, so that's how that would work. All right, lab on a chip. This is just a concept that we have like the availability and of all these simple tests like this, and we can compile all this stuff on very tiny little literal like chips that we can plug into machines, and those machines can analyze so quickly that nowadays it just makes testing so much more mobile and versatile as well. Depending, on the disease we're looking for, but it has been quite useful, and the concept of it has also been quite useful. There are new chips that also employ CRISPR technologies in order for identification of conditions and things like that. We think that the biggest impact for these things as they are being developed and utilized in reality more and more, we think that the biggest impact, of course, is going to be in developing countries where diagnosis and the tools for diagnosis are not readily available. And these also won't expire as quickly as reagents. Okay, then we have mass spectrometry, like MALDI-TOF. I don't remember what MALDI stands for, and I apologize for that. But I know TOF is time of flight. But basically, what this does, we have ions that we shoot at a sample, patient sample, let's say, and then that will be analyzed by a machine. Their time of flight will be analyzed as well. And anyways, based on their reaction, their time of flight, how they are separated by the machine, and when they reach certain indicators and all that in the machine while it's being analyzed, and separated into ions and particles. Anyways, the machine is able to analyze those particles as they are made, and as they reach to the sensor, that's the word I was looking for, reach sensors in the machine, and that can help it determine what the molecular makeup of these things that you're testing would be. Wow, so that was really eloquently stated, but I'll just say it one more time in more clear words now that I have a concept, which is to say, you have a patient sample, you shoot a laser at it, you're going to get ions made, and the machine has sensors that will kind of measure the ability of those ions to move within that whole situation. And based on those reactions, it can determine what the molecular makeup is of that patient sample. Okay, you can also use this to detect certain antibiotic susceptibilities. Cool. All right. This is just showing some of the results that you might get based on what molecules you might have in the sample. You'll get certain peaks, and then that can be interpreted as certain actual molecules, like compounds and everything like that. All right, finally, we have imaging. This is quite simple. Whenever you go to get an MRI, a CT, PET scans, we can often use these to look at deep tissue infections and problems with certain localized infections. So we can look at it in more detail on a screen before we cut you open to start doing stuff, and also allow us to find what areas we need to maybe put a needle in order to take a sample or biopsy an area if there is an infection deeper in the tissue, so that it will be less invasive for the patient and much safer for them. So that's it. Explain to me why there are so many different diagnostic methods available when we could just use PCR. Why do we have all this other stuff still? Why do we even need to know about that? Why do we use that? So that's that, and I will see you for the next one. Goodbye.